Bismillahirrahmanirrahim. So, right, we're starting off with our classes for O-level and IGCSE biology. And what you see in front of you is the syllabus for biology 0610, which is the syllabus code for IGCSE biology, since the student we have currently is an IGCSE student. This syllabus is valid for 2026, 27, and 28. Excuse me. Right. And so we'll be appearing in 2027, so it's valid there. Scrolling down, you see the usual contents and why choose the syllabus and all that other detail. So just like all the other science syllabi, right, the syllabus aims, right, to enable the students to acquire scientific knowledge and understanding of scientific theories and practice, develop a range of experimental skills, including handling variables and working safely, use scientific data and evidence to solve problems and discuss the limitations of scientific methods, communicate effectively and clearly using scientific terminology, notation, and convention, which basically means when you're giving a science paper, you talk in science, right? That is your language.
Yeah.
Understand the application of scientific knowledge, that the application of scientific knowledge can benefit people and the environment, right? And it could benefit you as well, obviously, right? Physics, chemistry, biology, all of those, knowing those things will help. And enjoy science and develop an informed interest in scientific matters which support further study. So be inquisitive. This is the syllabus, right? These are the chapter headings, let's say. So you've got 21, right? In O-level, they're 19. So this one's not lengthier. They just have sections of the content under different headings. The content is pretty much the same. So characteristics and classification of living organisms, organization of organisms. So, right, O-level starts off with this, right? IGCSE starting off with characteristics and classification. Then you've got movement into and out of cells, biological molecules, enzymes, plant nutrition, human nutrition, transport in plants, transport in animals, disease and immunity, gas exchange in humans, respiration, excretion in humans, coordination and response, which will cover both humans and plants. Then you've got drugs, reproduction, inheritance, variance, and selection, organisms and the environment, human influences on the ecosystem, and finally, biotechnology and genetic modification. Right, you have the usual 6 papers, of which you'll be attempting 3. So for the right, you've got the core and extended. And since we'll be doing the extended papers, we're going to be doing paper 2, which is the MCQ, paper 4, which is the extended theory, and paper 6, which is the alternative to practical, right? So 2, 4, 6. And the paper 2, which is the MCQ, is 40 marks, 40 MCQs, just like for the other sciences, and it's 45 minutes long. It's worth 30% of your grade. The theory paper is 80 marks. It is an hour and 15 minutes long, which is 75 minutes, which is actually shorter than what Cambridge would usually give, right? It would usually give an hour per mark, which would give you an hour and 20 minutes at least, right? So probably because you have, like, matching tables and fill in the blanks and stuff like that, which doesn't take as long to answer.
Yeah.
And then you've got paper 6, which is the alternative practical. It's a 40 mark paper, 1 hour long, and 20% of your grade. These are the assessment objectives, right? They're split into knowledge with understanding, right? So using terminology and vocabulary and instrument and technique and all of that, handling information, so using data and manipulating it numerically, doing calculations, identifying patterns, reading graphs and stuff, and then experimental skills and investigation. So this is, right, there are like experiments that will be discussed in biology as well, and so, right, making observations and interpreting data and all of that stuff. Now, subject content here is where they're giving details as to right, what the syllabus has under each topic. And right, I'm not going to go into detail of that right now, but what I will mention is the usefulness of the syllabus. Right, so the first thing it's useful for is obviously as a checklist, right, for progress. So, right, when you are covering the chapter, when you're learning it, in this case, when I'll be teaching it, you can check whether all of these top concepts have been covered. Okay.
Yeah.
And obviously, since I'll be using the endorsed text to teach, right, as a reference book, these topics will be covered. But you can always go back and see, right, is this topic covered? And if not, you can ask, and I'll okay, fine. This is when we covered it, right? This is the concept behind this, and so that brings me to the second usefulness of the syllabus is as a revision checklist. So not only as a checklist for progress, but a revision checklist that you can go and you can see, oh, this topic's covered. That's the concept, and so they're all they're not like mutually exclusive points. And the third one is as a summary, right? So we can use it as a quick, well, let's say, right, I'll write as a summarized revision guide, right? So you can use it for quick revision as well. Now, how does that work, right? So you can see that the way they're describing the characteristics of living organisms is that movement as an action by an organism or part of an organism causing a change in position of place. So they're defining what movement is according to the syllabus, right? They are defining what respiration is. It is a chemical reaction in cells that breaks down nutrient molecules and releases energy for metabolism, right? Excretion is the removal of the waste products of metabolism and substances in excess of requirements. So they're not only telling you that you should know this, they're also telling you that this is what you should know. Right? Did you get that? You there?
Yes, sorry.
Yeah. So that's why I say that you can use it as a revision guide, a summarized revision guide. Obviously, it's not. It's missing the details. It's missing the explanations. But yeah, and right, if you are like 100% on the syllabus, then right, you are very well good to go. Obviously, practicing the past papers and how to attempt the questions is a separate skill as far as the content is concerned, right? Knowing all of this. So skipping forward to the other stuff, but again, I'm not going into detail of the content because we'll cover that as we go. You can, and I guess you should read it on your own, at least maybe like the first chapter. So as we are going chapter to chapter, read in advance that this is the stuff that we'll be covering. Okay, details of assessment, we've already done that, so you don't need to take a look at that again. Yeah. So now what they're mentioning is, let me, where's the person? Okay, there it is. So the practical test and alternative practical require the same experiment skills to be developed and learned, right? So what they've done over the the recent update, Cambridge has sort of aligned the practical and alternative practical. So in a practical, the student will be standing in a lab and conducting the experiment following the instructions, whereas an alternative to practical, It's going to be the same experiment. You're not going to be conducting it. It's going to be on paper. So they're going to explain, this is the experiment, this is what the student does. And using your knowledge of practicals, tell what, and what. Okay?
Okay.
So they're aligned. They require an understanding of the same experimental context, and they test the assessment objective A03, obviously. Candidates are expected to be familiar with and maybe asked questions of the following experimental context. So, simple quantitative experiments like measuring volumes and masses and temperatures and all that stuff. Diffusion, osmosis, food tests, the rates of enzyme catalyzed reactions, including judging endpoints such as color changes, pH, and the use of hydrogen carbonate indicator, litmus, and universal indicator. Photosynthesis, right? So we'll talk about photosynthesis and measuring the rate of photosynthesis and limiting factors for that. And obviously, at this point, all of this might seem foreign or maybe very, very faintly familiar, right? But we will go through this in detail, right? As much detail as the syllabus requires. So you've got transpiration, heart rate and breathing rate, respiration, tropic responses, observation and dissection of seeds and flowers, germination, continuous and discontinuous variation, sampling that are representative and avoid bias. So, right, considering sampling size and random sampling and stuff like that. Observe, record, and measure images of familiar and unfamiliar biological specimens. Make clear drawings. So that's a skill you need to have. You need to be good at making sketches, not detailed sketches like outlines. So make clear line drawings of biological specimens, calculating the magnification or actual size, right? Labeling them. So this is all stuff that you will be taught, obviously. Excuse me. I had a bit of the throat's scratchy. It's like the season or something.
Yeah.
So use simple apparatus in situations where the method may not be familiar to the candidate. So like, okay, fine, this is the situation. I haven't done this in class. I'm familiar with the concept. So what can I do? All right. Then candidates may be required to do the following. They might be required to demonstrate knowledge of how to select and safely use techniques, apparatus, and materials. Right. And so identify apparatus, draw, complete, and label diagrams, explain techniques, appropriate apparatus, tests, pH, explain hazards, explain techniques, plan experiments and investigations, right? So we're familiar with that. Identify the independent and dependent variable, explain how the variable should be kept constant, appropriate number and range of values for the independent variable, the most appropriate apparatus and technique, experimental procedures, identifying risk and suggesting safety, how to record results, how to process results, and prediction of expected results. Then make and record observations, measurements, and estimates. So take readings from apparatus, take sufficient observations or measurements, including repeats and replicates. Record qualitative observations from food tests, so maybe a color change, maybe something like that, qualitative. Record observations and measurements systematically on a table, something, maybe drawing a graph. Right? Interpret and evaluate experimental observations. So processing the data for further calculations, of graph plotting, using a calculator as appropriate, presenting the data graphically, analyzing and interpreting observations and data. Again, including graphical representations. Use interpolation, extrapolation. So interpolation is, looking at the data that you have, and extrapolation is sort of making a guess of how the data would look like for points that you don't have, right, beyond the experimental values, right? And so you're going to do that using a graph and a gradient or intercept. Form conclusions justified by your observations, and evaluate the quality of the observation data, right? So is my data reliable? Is it correct? Is it, is there something wrong with it? If there's something wrong with it, then talk about it. Evaluate methods and suggest suggest possible improvements. So evaluate experimental arrangements, methods, techniques, identify sources of error, suggest possible improvements to the apparatus or the experimental arrangement or the technique that was used. Right. So far, so good.
Yeah.
Then comes apparatus, materials, and reagents, right? So the stuff they're mentioning over here is stuff that you should be familiar with. Okay. Now, the list is not as long as obviously in the syllabus for chemistry, so we can breeze through this. So the chemical reagents, right? First of all, are the safety labels. So C is corrosive, double H is health hazard, F is flammable, N is hazardous for aquatic environment, MH is moderate hazard, T is acutely toxic, and O is oxidized. All right, so those are hazard labels. Then the reagents you should be familiar with for biology: the Benedict's solution, the biuret reagent, carbohydrates, proteins, and lipids, something called DCPIP, dilute acid, dilute alkali, distilled or deionized water, enzymes, ethanol, indicators, such as universal indicator or something else, hydrogen peroxide solution, iodine in potassium iodide solution, lime water, methylene blue dye, petroleum jelly, such as Vaseline, sodium chloride, which is basically just regular salt, and sodium hydrogen carbonate or sodium bicarbonate, right? So it's known by two different names: sodium hydrogen carbonate, sodium bicarbonate. Don't get confused by one or the other; they're both the same. The apparatus that you need to be familiar with, and this is going to come in. This is going to be handy in paper 2 and paper 4, but obviously in paper 6.
Yeah.
Right? So a balance to measure up to 500 grams with precision. Right, so beakers of various sizes, a balance to measure up to 500 grams with a precision of at least 0.1 gram. So they're telling you, right, this is like the maximum you might have to measure, and you need a precision of at least 0.1 grams, right? So you're not going to take like a regular weighing scale, which goes up till like 160 kilograms. You're not going to need that. Right, and then it's only accurate up till 1 kilogram, right? It doesn't get more accurate than that unless it's a digital scale. Again, we don't need that kind of thing. So the thing we're talking about is a laboratory balance, right, or a weighing scale. Beakers of various sizes, such as 100 centimeter cube, 250 centimeter cube. It could be smaller than that as well, right? Bunks to fit standard test tubes and larger test tubes. This is boiling tubes. Bunks for delivery tubes to fit standard test tubes and with delivery tubes to fit standard test tubes and large test tubes. Filter funnels, filter paper, forceps, glass rods, hand lenses of at least ×6 magnification, lamps, photosynthesis experiments, means of cutting biological materials, so scalpel, right? You're not going to use a knife and cutting board, scalpel and sharp knives. Right, means of writing on glassware, so water resistant markers or wax pencils, measuring cylinders again of different volumes, mounted needles or seekers or long pins with large heads, rulers graduated in millimeter, scissors, partially permeable membrane, which is right. So how you're going to get that? So it's whisking or dialysis tubing, and you can see whisking is like a brand name. That's where it's capitalized, and you got a registered logo, like symbol over there.
Yeah.
Pasture or dropping pipette, petri dishes, spotting tiles, stop clocks reading to 1 second or better. Right, so maybe better than 1 second, split seconds. Syringes again of various volume. Test tubes, right? So the standard 125 by 15 millimeter and larger ones, which are 150 by 25 millimeters. Test tube racks and test tube holders. Thermometers going from -10 to 110 degrees Celsius with 1 degree centigrade graduations. Wash bottles, white tiles, or other suitable cutting surfaces. So you're going to cut using tiles, not a cutting board. So that was all about apparatus and chemical reagents. Then you got numbers, right? So the math involved in biology. You need to add, subtract, multiply, and divide. I'm hoping we can do that properly.
Oh yeah.
Yeah.
Okay.
Right.
Yeah.
Using decimals, fractions, ratios, and reciprocals.
Okay.
It not something you really often need to use, but still, right?
The
Can
basic.
yeah,
Basically.
the basic stuff. Calculate and use percentages and percentage change using standard form, which is something I hope we are well versed with. Express answers to an appropriate or given number of significant figures. Express answers to an appropriate or given number of decimal places. Rounding answers appropriately. Algebra, recognize and use direct and inverse proportion. Solve simple algebraic equations for any one term when the other terms are known. So finding an unknown basically. Substituting physical quantities into a formula. So that's the algebra. Geometry and measurements, converting units, understanding the meaning of angle, curve, circle, radius, and other terminologies given over here. Recall and use equations of the area of rectangle, triangle, and circle. Recall and use equations of the volume of rectangular block and the volume of a cylinder. Use a ruler. Make estimates of numbers, quantities, and lengths. Understand surface area and use the surface area to volume ratio. Use scale diagrams. Select and use the most appropriate units for recording data and the results of calculations. What this means, if you're measuring length, if it's not something very long, you can measure in millimeters. If it's something far longer, which is not something you'll be doing, but still, then you can do it in centimeters. Right?
Yeah.
Graphs, chart, and statistics. So draw charts and graphs from data. Interpret line graphs, bar charts, pie charts, and histograms with equal intervals. Interpolate and extrapolate from data. Determine the gradient and intercept of a graph, including units where appropriate. Select suitable scales and axes for graphs. Recognize direct and inverse proportionality from a graph. Calculate the mean and range of a set of values. Use simple probability. There's still a bit, not a lot. So presentation of data, right? Taking and recording readings. So the data should be, this is something that we need to be extra attentive about, right? It's going to come into practice obviously later, but if we just focus on it a bit right now, then and we come back to it again later, so it won't be new to us at that point. So data should be recorded so as to reflect the precision of the measuring instrument. Right? For example, the smallest difference that can be reliably detected on the measuring instrument scale should be reflected by the number of decimal places given the measure. What this means, right? If I just show my ruler, right? Or if you have a ruler. So this thing over here, right? And
Okay.
yeah, I don't know. I switched on my camera for you, but not loading for some reason.
Yeah.
Anyway, if you have a ruler with you, right, and you take a look at it, then you can see that the smallest measurement that you can make is 1 millimeter, right?
Yeah.
And so what you would expect is, for example, if something is 2 centimeters and 7 millimeters, and so what you would get is 2.7 millimeters, right? So the decimal places to show the most reliable, the most accurate thing that you can get out of this, right?
Yes.
That's what they mean. Oh, and the reason Google Meet isn't able to access the camera is because it's being used by the recording software.
Oh, okay.
Anyway, if in the future something that I absolutely need to show to you, then obviously I'll switch.
Yeah.
Okay. So a measurement or calculated quantity must be accompanied by a correct unit, right? So if it's millimeters, then you need to add millimeters. If you're writing centimeters for millimeters, that's going to be wrong. Each column of a table should be headed with the observation or physical quantity, right? So if it's length, right, and then you put a slash, which is called the solidus, right? The forward slash, I believe, what we call it normally, and then the unit. So for example, time in seconds, right, or length in millimeter,
Yeah.
right? And so this is to be used for separating quantities and the unit in tables, graphs, and charts. Unit should not be included with data in the body or table. So what they mean to say is that if you put the unit in the heading of the column or row, whatever the case is, then you don't need to rewrite it with every value you're putting in, because it makes sense, right? Anything that's under that heading is of that unit, right? If you're measuring time in seconds and you go like, oh, 2 seconds, 5 seconds, 8 seconds, you don't need to keep writing 2s, 8s. Like, right? I do have my pen with me. So, right? If this is the column heading, then the only thing you do is type 2,
Just write the
5,
numbers.
8, 11. You don't need to write 2s, 5s, y, because you already put the unit in the heading. So you don't need these values, right?
Yeah.
It is understood that if it's 2, right, that means it's 2 seconds.
Yeah.
The data should be recorded to the appropriate number of significant figures. So that depends on the level of accuracy of your readings and, right, appropriately whatever calculations you're making. Graphs. The column headings of a correctly headed table can be directly transferred to the axis of a constructed graph, right? So for example, time and, right, let's say, what are we measuring?
Millimeter.
No, something to do with time. And yeah, so let's say, like,
Hour.
yeah, no, bubbles, right? Number of bubbles, for example, right? Conducting some sort of experiment. And so what we do is when we make a graph out of this, right? And so we're going to take this and put it directly on the axis. So this is times per second, and this is,
Hours.
right, number of bubbles. A graph should be drawn with a sharp pencil, right? Not a blunt pencil, no thick lines, no broken lines, no overwriting. A graph, right? Each axis should be labeled with the observation of physical quantity and the unit where appropriate. Unless instructed otherwise, the independent variable, this is very important. The independent variable should be plotted on the x-axis, right? So the stuff that you are changing in the experiment, right? If
So
that is some,
bubbles is the dependent value because it depends on the time, right?
it depends on whatever we're changing to get the bubbles, right? Yes, right. So if the independent variable is something that you're putting on the graph,
Okay.
right? So for example, let's assume time is the independent variable, because that's like, okay, fine, how much time are we giving to this reaction or whatever it is that we're conducting a practical about, an experiment about. Then it goes on the x-axis. And whatever the dependent variable is, so whatever the observation you're making, whatever the, I wouldn't use the term result, but whatever like change you are observing in the experiment and you're making notes of it, right, that is the dependent variable. And so that goes on the y-axis. So unless instructed otherwise, if they're telling you no, do it this way, then you do it like you're being told. If not, then the independent variable goes on the x-axis, the dependent variable goes on the y-axis.
Yes.
Unless instructed otherwise, the scales of the axis should allow more than half of the graph grid to be used in both directions, right? So how do I show this? Right, if this is your page, well, this is your graph actually. right? This is your grid. And so you need to use, right, about this much also, at least, right? So you need to use more than half of the graph grid, right? So at least
Yeah.
this much should be used. A safe way of going is using at least 70%. So let's say using this much, right? Of course, I'm
Yeah.
just like, this is just for explanations, not an actual 70%.
Yeah. Yeah.
Right now, obviously, depending on whatever values you're giving, the scales can be different on the x and y axis, right? They don't need to be the same, but they should be in sort of increments that are easy to work with. So they're mentioning over here, right, in increments of 1, 2, or 5, because 1, 2, and 5 is easy to work with, right?
Yeah.
4 is a multiple of 2, so that's also easy to work with depending on the data that you have. Don't take something like 3, because when you start dividing 3, then you're getting like, oh, it's right 1.5, and then it's 0.75, and it becomes difficult,
Yeah.
right? Of course,
Yes.
we, depending on the values, we can have a graph that is using about 90% or more of the graph, right? So something like this. So it's going like it's utilizing the maximum area, and that's what we want.
Yes.
Okay, the axes do not have to include a starting point of 0 and 0, right? So the graph for the x-axis can start at maybe 10, and the graph for the y-axis can maybe start at 5, right? So it doesn't have to be 0, 0. the the scales and the labeling of the axis in that term are independent of each other, right? One of them can be 0. For example, it could be 0 and 10, or it could be 5 and 0, right? Whatever the case is, or 0 and 0 if that works. Points on the graph should be clearly marked with crosses or encircled dots of appropriate size, right? So of course, when you come to doing graphs in the paper, right, we'll go over that again. Each data point should be plotted to an accuracy of 1/2 of the smallest square on the grid. All right, so maybe if a grid represents 1 unit, then half the smallest square is like 0.5, and so that's the accuracy that should be. A best line should be a straight, should be a single, thin, smooth line or curve, right? So a best fit line, when they're saying a best fit line, it does not automatically mean straight line. It could be a curve,
Yeah.
right? It should be single, so no overlapping, no like, oh, right, no, it's going double like that, no overlapping, no broken lines. It should be smooth. The line does not need to coincide exactly with any of the points. So, right, unlike the lower class, you're like, oh no, it needs to go through the point. No, it does not. Because the values you get in actual experiments are not the kind where it, the line is passing exactly through the points, right? So you can have something like maybe like this, and so
And it can
you
go
it
opposite the dots.
so it's going to go like, right, a line of best fit sort of is drawn where, right, the number of points, I wouldn't say like the number of points are equally distributed, but like when you draw it, it's like, okay, fine, right,
They're
it's,
closer to the line,
yes,
basically.
they're close enough to the line. The line is sort of like, if you were to draw a line, this is the kind of line you would get, right? It is
There's a
justified.
name for
They,
this
they
in maths. Uh,
right, that's what's called a trend line over here, as you see, right? So this is what the line is showing, is basically the trend of the results. It's not the actual data points,
yeah.
right? And this is perfectly fine. Now what you need to do, obviously, is if you can make this any better, then you do that. Okay, so the line does not need to coincide with any of the points. Where there is a scatter evident in the data, examiner should expect a roughly even distribution of points on either side of the line over its entire length. So something like this, like you have 3 points below the line, 3 points above the line, works. Maybe I can adjust the angle of this thing a little bit. Maybe I can draw it like that. I don't know, but right, if you can evenly distribute the points above and below the line, that's good. Points that are clearly anomalous should be ignored, right? So if you got a point, for example, over here, you're not going to worry about that point. Like, oh, how do I make a line of best fit? Because like, no, obviously there's something wrong with that.
Yeah.
A best fit line or curve should only be drawn if there's good reason to believe that the intermediate values can be predicted. Right. So if this kind of an experiment where you're like, no, I can't make a prediction because right, something changes. For example, the temperature changes and the enzymes become denatured, and so you're not going to notice an increase in enzyme activity, you're actually going to notice a fall in enzyme activity. And so over there, right, you might not be making a line of best fit. Candidates should be able to take readings from the graph by extrapolation, right? So for example, this is still where the graph has been drawn, right? So till here, let's say.
Yeah.
Right? And so what I need to do is I need to find something that is right of a smaller value, which I did not do in the experiment. And so what I will do is I will extend the line and then I will take a reading of the graph. And that is called extrapolation. It can work for lower values, it can work for higher values as well. Okay.
Yeah.
Okay. So extrapolation, interpolation is basically I've got this line with the data points, right? So maybe this data point is 4 and this data point is 16. And they're asking me, oh, what happens at 10? I don't know what happens at 10. I didn't take that value. Well, use the line. 10 is somewhere over here. Use that.
Yeah. Okay.
Data values should be read from the graph to an accuracy of 1/2 of the smallest square on the grid. So we talked about that. Drawings should be drawn using a sharp pencil to give fine lines, right? So if I'm drawing right a tree and I'm not good at drawing, I don't know how I survived O-level biology, right? But I did, right? Maybe I was better at drawing back then. And so this is how I still draw trees, right? And so they should be fine lines, clear and unbroken. You can obviously see my line is wobbly over here. So what you would want is like a nice smooth line. And so you need to practice your strokes, right? Drawing should use most of the available space and show all the features observed in the specimen with no shading, or so you don't need to shade. I don't need to go like, oh, this is a darker shade of green, right? And this is like, pat. You don't need to do that.
This is not an art class.
It's not art class, exactly, right? So what you basically need to do is draw an outline. If there are certain features that are given in the image that is on the paper, then you need to represent those features in your sketch as well, right? And label lines should be drawn with a ruler and touch the object or feature, right? So for example, right, this part, right, is, and the label, yeah, and so this is the trunk, and the labeling, the text is always horizontal, right? And this is the canopy.
Canopy.
Yeah, I believe if I'm right. And another thing is, for example, right, you're not going to have the lines, oh, look, right, roots are over here. And so I'm taking this line and going, oh, crossing it through the other line and going roots. No, right? No crossing of label line. So you write it separately, roots, for example, or whatever it is.
Yeah. Okay.
Okay.
Basically, everything has to be neat and clean.
Yes, basically. Proper. So charts. Pie charts are generally used to show percentage and proportionality. Bar charts should be drawn for categorical or discrete data. They should be made up of bars of equal width that do not touch. So, right, if you have a bar chart, right? And right, so there's a space in between the bars, and they're discrete data. For example, colors. So colors, you've got R, B, G, red, blue, and green, right? And
Yeah.
histograms should be drawn for continuous data, right? So heights, for example, heights of people is a very standard example. And so continuous data, that. And then in that case, the bars are touching. Ah, that's a lot, huh?
Yeah.
Okay. Is there anything? Yeah, nothing really of particular value. What you do, avoid confusion concerning the symbol for liter. The equivalent quantity for cubic decimeter will be used in place of 1 liter. Right. So wherever there's like liter involved, right, they're going to use decimeter cube.
Okay.
Right. So 1 decimeter cube is 1,000 ml, which is 1 liter. I think the final thing, the final thing that we need to talk about are command words. Now, obviously, these come in handy when you're doing the exam. But again, since we're going over the syllabus, then might as well take a look at it. So when they're asking you to calculate something, you need to, work out, the figures and the information, and do some workings, to do some math, and then give an answer. Compare.
Yeah.
So identify or comment on the similarities, or compare goes both ways, similarities and differences, or differences, like whatever the case may be. Define, give precise meaning. If they're asking you to define something, you need to give precise meaning. Describe, state the top points of a topic, or give characteristics and main features. Determine, so establish an answer using information available. So they've given you a table and they're asking, oh, determine what is happening. And so you're going to use the values in the table and then give your answer, right?
Mm-hmm.
Or whatever other information that they're giving. Evaluate, so judge or calculate the quality, importance, amount, or value of something, right? So evaluate so and so thing. And so depending again on the question, you're going to say this is like this is like that, right? So and so. Again, right now we just like we're not looking at a paper and we don't have that kind of question in front of us, so it will make more sense when we are actually looking at a paper. Achha, and
Okay.
yeah, explain. So you make relationship between things clear, say why or how. You need to give evidence to support your reasoning. Give, when they're asking you something like give, and then so you just need to, reproduce something from memory. Right? They're going to like give the reason why this is, and so you need to recall that concept and okay, fine, this is the reason that they gave in the textbook, and you write down that reason. Identify, so you like select something, you recognize something from the information, you name something. Outline, you give the main points. Predict, is suggesting something based on the information available. Okay.
Okay.
Sketch is, as we just went over, making simple freehand drawings showing key features, taking care over proportions, right? So if, for example, if I just go back and right, if the actual tree is much, has a much thicker canopy, then I need to draw it that way, right? So
Yeah.
right, this would be incorrect. So the proportion would be like, oh, it's like has a very thick canopy, and so that is how I'm supposed to draw. Okay.
Yes.
And so whatever other features are given, larger or smaller, and a particular angle. State means expressing something in clear terms, and suggesting, again, applying knowledge and understanding to situations where there are a range of valid responses. All right, so you're making one suggestion. There could be other suggestions, possibly. Because again, this will make more sense when we are done with the syllabus and come to practicing past papers.
Yeah.
Right, and yeah, that all we need to know from the syllabus. Right, we're done with that. So
Okay.
yeah, next class, inshallah, we will start with chapter 1, which is what again?
I think something related to characteristics.
Characteristics of organisms.
Yeah.
So here, yeah, characteristics and classification of living organisms. All right.
Yes.
Any questions? Anything you'd
Well,
like to share or add or whatever?
well, nothing at the moment.
Okay. So come prepared tomorrow, right? Have have writing material with you to make notes, and be attentive, be involved, and be inquisitive.
Yes.
All right.
Okay. Yeah.
All right then. Tomorrow, inshallah.
Inshallah.